Okay, so here we are going to understand the Lowry assay. And the Lowry assay, it is going to be similar to the Biuret assay, so I would encourage you to watch the Biuret assay video first before jumping into this one. But to quickly refresh our memories, remember that the Biuret assay is made up of the Biuret reagent. Now the Biuret reagent has three components. It has copper sulfate, and the copper sulfate is where we are going to get our copper ions from, which play a very important role. Then we have NaOH, or we can have KOH, which is going to provide our alkaline medium. And then we have potassium tartrate, which is stabilizing those copper ions. But what happens specifically? So where does the name Biuret come from? Because the answer lies in the name itself. So Biuret is actually the composition of two urea molecules. So essentially, when we heat up two urea molecules, it forms Biuret, and Biuret is just a composition of two urea molecules. We can actually break down the name. Bi means two, and then uret for urea. So Biuret is just two urea molecules. And the reason why Biuret is so important is that it mimics that peptide bond that is found within proteins. We can see that over here. We have our carbon with our double bonded oxygen and our nitrogen with that hydrogen. Now, since it mimics that peptide bond or amide bond that is found within proteins, initially when we were studying this form of assay, what we did was we added in copper ions with Biuret. And we noted that the copper ions actually complex with the amide bond within Biuret, and that complex absorbs light, and through that we can quantify proteins. So essentially, the first step of the Lowry assay is taking that Biuret assay approach, in which we are actually utilizing proteins. And remember, proteins are long chain peptides, and they're made up of amino acids, and the amino acids will have variable R chains. Now, what's important to note is that between each amino acid, we are going to have a peptide bond, and at that peptide bond, we will see the interaction of copper. Now, copper will go on to interact with four different peptide bonds, or four different nitrogens, and we get this complex form, and this complex will absorb light. But what makes the Lowry assay better than the Biuret assay is that this copper, which has been reduced because it's interacting with the peptide bonds, this copper can now go on to reduce another reagent, which is called the Folin reagent. Now, the Folin reagent is an oxidizing agent. What does oxidizing agent mean? Oxidizing agent means that it's going to oxidize something else, and it itself is going to be reduced. Now, looking at the Folin reagent, it actually has these two subcomponents, but essentially what happens is that the overall reagent will be reduced by this copper. Now, we have our reduced copper's ion, and then once it reduces Folin, it's going to be oxidized into this cupric 2+ ion. So essentially, the theory behind this assay is that the more proteins we have, the more amide bonds we have. The more amide bonds we have, the more interactions we can form with copper, because remember, copper forms those interactions with the amide or nitrogens found within our protein. And the more interactions we have, the more intense purple color we will produce, and as a result, we will get a higher absorbance value. So what we do is we take our sample, and then we add in the Biuret reagent, the Folin reagent, and then we can place that sample into a spectrophotometer and then observe the absorbance values. And the greater absorbance value will mean more proteins. So to quickly recap, what is the Lowry assay? Well, we can divide it into two steps. The first step is actually going to be the interaction of copper ions with the peptide bonds of our protein. And then the second step is that copper ion reducing the Folin reagent. And as a result, we can understand the more proteins that we have, the more amide bonds, the more interactions with our copper, and then the more interactions with the copper will give us more reduced Folin, and then we will produce a higher, more intense purple color with a higher absorbance value. And that is the Lowry assay.